Uh, Hi, everyone. So it looks like Dr. Circle's talk has not ended yet, so I'm kind of like continuing that. So my name is Sunandita Sarker. I recently joined as an assistant professor at University of Massachusetts, Amherst, and I'm just finishing up here with Dr. Sokol in his bio-inspired advanced manufacturing lab. So today I'm going to talk about some of the soft robotic applications that he just talked about a little bit. And my project is about a soft robotic biopsy tool that's specifically designed for pediatric applications. So congenital heart diseases is actually a problem with a lot of children, and especially children born with like defects and abnormalities in their heart, and it could get pretty complex situation where the only treatment option available is a heart transplantation. So heart transplantation in only in US, about 12% of all the heart transplantation is in pediatrics. So a major part of heart transplantation, like after basically the surgery, is like periodic biopsies of the allograft or the transplanted heart to make sure that not like for symptoms for rejections or how the grafted tissue is functioning. So the process of a cardiac biopsy or endomyocardial biopsy is they insert a guide wire and then a sheet to the target location, and then they insert the biopsy tool that following that guide wire and take a small piece of the tissue from the basically from the heart. So here there are certain challenges, like Dr. Sokol mentioned previously a little bit. Like some challenges are definitely that these are done under X-ray, so exposure to radiation, and also there's like some other obstacle with that. And because there's a very limited maneuverability control, so there's some unintentional injuries as well as the manual bending, like to reach the target location, doctors or medical professionals, they bend the tool by hand, and that really like limits like in which locations the tool can reach. There are certain parts of the heart that is completely not like reachable with current state-of-the-art biopsy tools, such as the left ventricle and left atrium. So soft robotics could be a possible solution to this kind of challenges. And in recent years, we have seen quite a bit of interest in using soft robotics in surgical applications, more specifically for biopsy applications. For example, there are researchers, they are using soft robots for like lungs biopsy, also like gut biopsy, and other application where size is like you have quite a bit of room to work with. So basically they have great dynamic bending, so you can control the navigation, and as well as it has like a less less unintentional injury with this kind of devices. Recent work, it's also been introduced in like cardiac applications, where like a control catheter with different kind of like surgical tool attached to it could be used for like cardiac interventions. But we still have some challenges left with this type of tools. And one of them is like there's a lot of metal components using in these soft robots, and that kind of limits our options to like X-ray guided only, and which could be like very harmful for especially like pediatric applications. And the major thing that is that the scale. So it's still in the millimeter to centimeter scale. And for some application, you need something that is like even sub-millimeter, especially for like pediatric, like heart surgeries, especially for like neonates or even preterm babies. It could be like less than a few hundred microns. So the goal is to use additive manufacturing or more specifically, direct laser writing enabled additive manufacturing to make these micro-scale soft robotic biopsy tools that can be used for pediatric myocardial biopsies. So some of my prior work with additive manufacturing is like a hybrid additive manufacturing that we did, where we combined two different type of 3D printing technologies to make biomedical devices. For example, here we may use digital light processing or DLP to make some capillaries for stem cell therapies. And we use direct laser writing to build 3D, to build nanostructure on top of those capillaries that can be used. So the direct laser writing process uses a pulse, like a tightly focused infrared laser that scans through the liquid material and polymerized only the designated location to get the 3D structure. So here it's 3D printing a microneedle array on top of microcapillaries that can be used for like microfluidic application. So this is the fabrication process of directly on top of the 3D printed capillaries, and you can see like you can get a very large geometric variations from this process. So we work with our collaborators in the neuroscience department in University of Maryland, Baltimore, to do some injections in mouse. So my next few slides has like tissues and blood. So if you're not comfortable with that, you can avoid the next part. But overall, actually, my rest of the slides will have some sort of biology because I'm working with like biomedical applications. So we excised the mouse brain from this mouse, and then we did like an injection experiment to see if we can inject some fluid into the brain using our 3D nanoprinted microneedle array. So in the next video, here we're puncturing the brain tissue, and then we will inject some surrogate fluid, which is basically dyed water, into the surface of the brain. The idea is later on can inject suspended stem cells to help regenerate the damaged tissue of the brain. And what we saw is like with this 3D printed material, we were successful to like puncture the tissue surface and inject the fluid inside. Next, the other concept that Dr. Sokol just mentioned a little bit is the ex situ direct laser writing, where we are 3D printing surgical components directly on top of some kind of catheter or capillary that can be used for like catheter-based application. So in this one, what we are using is like we're 3D printing microneedles directly on top of like capillaries to use for like IVF or different kind of embryo studies. So we're here, we are using 3D printing needles to inject materials inside a fish embryo. So what currently I'm doing is kind of extending this ex situ direct laser writing strategy to 3D print a biopsy tool directly on top of a capillary or a catheter. So once this biopsy tool has two components. One is the soft part actuator that can inflate with pressure, and then the needle part can use for like tissue biopsy. Here's the fabrication process. So it's directly printing on top of the capillary, and we check for the adhesion of the material, the substrate material, which is a catheter or a tube on, and the biopsy tool on top. And you can see there are the three components of the tip of the biopsy tool. So next, we did some analysis of like the different designs of the actuator to achieve that kind of extension we would need for a biopsy application. So we try different type of cross-sectional geometry, and with and to see like how much of an extension we could get from this soft actuator. And we can see some pretty low extension, which might not be very helpful for an dynamic process of injection. So we tried to investigate other materials. So first we tried with like IPDIP2, and then we are also trying. So these are some commercial materials that is suitable for the 3D nanoprinter that we are using, but there are some other materials as well to kind of investigate and see like where we can find the perfect material and the geometry combination that could give us like the functionality is needed for this application. So based on that, we kind of chose the polynomial design to kind of go for our experimental study. And that's what we did, like a pressure versus deformation study. And you can see it's kind of following the computational model that it has a very low elongation with pressure. And overall, at a very high pressure of like 600 kilopascal, it has about 5% of elongation. So yes, that's not very great, like how you're going to puncture a tissue if you are only increasing it by 5%. But what another thing was important, like, is this material and 3D printed part is actually able to like puncture through any tissue or at all. So the next step was to check for that. So first we did like an in vitro study with agarose gel, which is very standard in the field to test, to compare like tissue properties. So we use like 10% agarose gel that is very stiff and very comparable to like tissues like a hard tissue. And we see that like with this needle was successful to puncture and did not have any break or damage to the needle itself. So next we move on to an ex vivo study where we used bovine heart, which is kind of funny because I got it from a grocery store to test like if this 3D printed needle is actually able to puncture through cardiac tissue or not. So here's the video of that. And like heart tissue is actually one of the very strong tissue in our body. And so that was a big kind of like a question mark, like proof of concept, like will this needle even break down or what's going on? So I was surprisingly happy that it was able to puncture and kind of leave like a small mark on it, but there's like room to be working, room to improve that overall puncture because like especially when it's like a live tissue, it's like it deflects very easily. So you need to be very sharp and kind of fast to kind of puncture through the first layer of tissue. So I talked about like this one all had like only 5% of elongation. Is that gonna even work? So then we move on to the next idea, like what if we do like a multi-material approach, which is pretty doable in a large scale, like the multi-jet printing or even extrusion, but it's kind of not very easy to do with DLW. So but I still had to try something. So we tried with the multi-material approach where we print the actuator with soft material, which is IBPDMS. And then after the actuator was printed, we switch out the material and we introduced a new material, which is a rigid material and IPDIP, and we printed the needle part with the rigid material. So now we have a dual material device that has a soft actuator and a rigid needle tip on top. So, and this, like as I mentioned, like it's multi-material approach for DLW has been done before, but for functional application, especially where force is a big factor, we need to make sure the interface between these two materials is strong enough. And from our observation, we see pretty strong adhesion between the polymerized IPPDMS and IPDIP, and they held well pretty good even after applying forces. So next is it helping with the elongation problem that we were saying before? So here you can, if the pressure is actually 1 magnitude lower than what we have even used before, and we have pretty high elongation, so like 30%. So we got from 5% to 30% by switching the material. Now, there are a lot of room of improvement. Like we can work with the geometry to get into like more elongation, like maybe 60% or 70%. And then, and also we can do it like pretty fast, so it can puncture through the tissue. So that's where we are at now. And so to conclude, so we have introduced this like new 3D printed multi-material and single material biopsy tool at an unprecedented like length scale, very small enough to be applicable for like pediatric surgeries. And both materials have shown like some sort of elongation. And especially with the dual material approach, we can get up to like 30%, and which can be improved on. And we have also seen like, yes, with 3D printed biopsy needle, you can puncture tissue and then retrieve from the tissue like safely. So my next approach is with this one is first, can we get like actuate-based puncture? And it's in a work in progress stage. So we are trying to get like a fast movement from the needle tip using the actuator to actually puncture the tissue. And then the next step is also the control and navigation. So this is, I'm also trying not just based on fluidic circuitry, like Dr. Sokol mentioned before, but by changing the geometry in a single tubing structure, but having like a designed like thickness or designed features, we can get different type of navigation capabilities with catheter-based surgical tools. So with that, I'd like to thank all the basically the funding resources and also the members of BAM Lab that helped me with my work here. So any questions for